All right, so I was very rudely cut off by Microsoft Stream, and I was saying that when you have a crossover, then those chromosomes are called crossover chromosomes. When you have that, those are going to be less numerous, and that's going to give you these smaller groups. That's what's responsible for these smaller groups in your data set. So this data set here that we see is pretty typical. And we're going to use this data set to figure out the distance between gene Y and gene W. Right. So we did the test cross exactly as you're supposed to, a double heterozygote cross to a completely recessive individual. And then in each of these cases, this chromosome on the bottom represents the chromosome that came from the father, either this or this. And then so the one on the top represents the one that came from the mother. And that's the one we're really focused on, the one that came from the heterozygous or double heterozygous mother, because we want to know how many or what the frequency of crossovers in meiosis was when the mother's cells went through meiosis. All right. So what we're going to do to figure out the distance between the genes, we're going to look at the groups of offspring and see which ones have parental chromosomes and which ones have recombinant. The way we do this is the largest group numbers have, by definition, they must have the parental allele combinations. And the smallest numbers have the combinations that are crossover combinations. How do I know this? Because the rule is for any pair of linked genes, in this case, gene Y and gene W, parental gametes or gametes with parental allele combinations will always be more common than recombinant gametes. And also, I gave you the mother's genotype in this problem, so you can see which ones must be parental allele combinations and which ones aren't. You won't always have the mother's genotype given, so usually you're just looking for what's the most common or most numerous type of offspring, and there should be two groups, and those are the two parental chromosomes or the two chromosomes from the mother, one in each of those large number groups. So we're going to calculate then the frequency of offspring with parental allele combinations and the percentage of offspring with crossover allele combinations. And so we add together the two biggest numbers and divide by the total. When I make up data, I use a kind of a nice round number, in this case 1,000. And so when we do that calculation, we actually get 0.995, but you convert that to percent by multiplying by 100, and that's 99.5%. When you do the percent of offspring that have a recombinant chromosome, it's 5 out of 1,000, which gets you to 0.005, but when you multiply by 100, you get 0.5%. So I do expect that you can go pretty quickly between decimals and percents in order to do this kind of work, right? So what we're calculating is the frequency of recombination or recombination frequency. That's really the thing we're focusing on here. So that second calculation on the last slide. And for short, we call that RF. And so in this problem, the frequency of recombination between the gray and the red genes, or between, I should say, the body color and the eye color genes, is calculated to be 0.5%. And that's going to be an important number because we use RF as the measure of genetic distance on the map. So here's the rule. If 1% of recombination frequency is equal to 1 map unit, so it's an exact correlation. So if we have 0.5% recombination frequency, then that's going to be equal to 0.5 map units. So you just get rid of the percent and you put map units, and that's the distance. The smallest recombination frequency that you could measure from a set of data is 0. That would be if you had no recombinant, no crossover offspring with crossover chromosomes. The largest number you could measure would be 50%. The problem with 50% is 50% could be an actual measure of distance, but it would be the same value you would also calculate if the two genes were not linked at all. So if you do calculate an RF of 50, it's not a useful measurement because it's really indicating that there is no linkage here at all. So you would not be able to convince someone that the distance between your two genes is 50, because it could just as likely mean that the genes aren't linked. If genes aren't linked, that means that maybe the eye color gene is on chromosome 2 and the body color gene is on chromosome 4. So 50 is typically, you're going to assume that means the genes aren't linked. So you would need to come up with some different kind of data if you wanted to prove they were linked. But right now, you'll just say 50 indicates non-linkage. That's going to be sufficient for us. So then you draw the map. So you draw just a general chromosome, just a line. And then you draw Y, just put it here, W here. You could put Y plus and W plus, that doesn't matter. But some versions, some allele Y, some allele of W, because different flies have different alleles there. But what's in common, what's always true would be the distance between them would always be 0.5 map units. And this is assumed to be an estimate, but it's considered to be a decent estimate. So about 0.5 map units between these two genes. It would not matter also if you put W here and Y here, because, chromosomes are not, there is no right side or left side. So if we rotate it around, it would still be 0.5 map units between them. So that does not matter. So when we are trying to map entire chromosomes, if you're mapping an entire chromosome, generally what you're going to do is you want to determine genes that are linked less than about 40 map units apart, and then combine or composite the maps to make the entire map. All right, so what I'm saying is a human chromosome, for example, is about 100 megabases, so about 100 centimorgans on average. And so, you can't map two genes at the opposite ends of a chromosome with a single set of data. You would have to map, gene A to gene B, and then gene B to gene C, and then gene C to gene D, and you would have to link together, no pun intended, you would have to composite together many maps to come up with a map of the entire chromosome. All right, so you can't do these large measurements, a single mapping cross, but you'll do lots of crosses to come up with smaller maps. Now, these maps are generally not done in human genetics for obvious reasons. You need large amounts of offspring from a single cross, and humans just don't provide that. So typically you would see mapping. This kind of mapping was done very heavily with Drosophila, fruit flies. All right, so we have a plant example. All right, we're back to this one. All right, so if you're going to calculate the recombination frequency from this data set, and it's not a very big data set, what you would do is, well, you can pause the video right now and see if you can do it on your own, but I'm going to work through it as well. So what you're going to do is the two smaller numbers represent the recombinant offspring. So you're going to add those together, right? Because we want to determine recombination frequency. And then you're going to divide by the total of all the offspring together. And then I need a calculator. So 55 plus 53 plus 8 plus 7 is 123. So 15 divided by 123 is 0.12195. So let's say it's about 12.195%. Get rid of the percent and put map units, 12.195 map units. I'm not too worried about how many decimal places you leave. There are rules for that in terms of significant figures, but I'm not going to teach significant figures, and therefore we'll just kind of round off. So if this was a test question, give me a couple of decimal places if it's an open-ended test question. If it's a multiple choice, pick the one that's close. And so that would be your answer for that. So about 12.195 map units if I did this right. Now, like I said, typically you would get a whole bunch of small map distances between genes, and then you would composite them together. And so this kind of problem is when you have those small map distances, now we're going to look at all of them and try to make a composite map, all right? The first thing to notice on a problem like this is, because here's where the RF is, so these are the map distances. If they give you a 50, that means something's not linked. So B and C are not linked, C and D are not linked. So I would ignore those for now. Ignore the 50s. They're not helping you. A and D are linked at 8 map units apart. A and B are linked at 5 map units apart. And B and D are linked at 13. So what I would do would be to start, my way of doing it is start with the biggest number. So you kind of put bookends on the map. So it doesn't matter if you put B on the left or on the right, but I'm going to do it like this. So that's 13 map units altogether. And then I have A to B is 5. So it could be here or it could be here. So A, I'm going to put a question mark. This is kind of trial and error. It's a little bit of a puzzle, if you like. And then A to D is 8. So if A was over here, then it would be 5 plus 13 would be 18 to D. So that can't be right. But if A is linked to B at 5, then A could be linked to D at 8, and those two pieces would add up to the whole of 13. So that's how you make the composite map. That's just a small example, but it's a little bit of trial and error and just making the puzzle pieces all fit with one map that's consistent with all of it. So this, like I said, this is called 2-point mapping. When you do a dihybrid cross to a completely recessive individual and you determine the distance between two genes. The problem with 2-point mapping is sometimes you will get double crossovers between the two genes, not very often, but sometimes, and they're not going to be detectable in the offspring because it really creates ultimately a parental allele combination. When you can only see the phenotypes of these alleles and you can't see the segment of DNA that's been swapped in between them, you will misinterpret these as being parental allele combinations. And so we say that with 2-point mapping, your recombination frequencies are not as accurate as they could be. And so that's why we do what's called 3-point mapping. And 3-point mapping minimizes some of the inaccuracies of 2-point mapping. I wouldn't say it eliminates it completely, but I should say it minimizes. I don't think you can eliminate, minimizes. All right. And so we're going to start looking at this kind of mapping in the next lecture segment. But really, 3-point mapping is 2-point mapping problems that are being solved simultaneously. So we're going to use the same logic to do 3-point mapping as what we did with 2-point mapping.